Yep. I'm live now. Oh, can you send me the link again, Manjula? Okay, we'll do one thing. I'm not going live. I mean, I'm not checking anything in my mobile. If there are any students in the live, you can just tell me, then I'll go live because I couldn't able to go live now. Okay. Uh, to whom I need to take a class? Yes. Anyhow, we'll go. I'll make you a co-host, and I'm not launching any polls today, as only host and co-host are in the class. Yeah. Yes, you are the co-host now, so we are fine to move. Yes. Hi. Good evening, everyone. So I just wanted to take a short class and a quick revision on the biotechnology. Okay, so let's get started without wasting a moment. Yeah. So the biotechnology. Hmm. Okay, sorry. So in this biotechnology, yes, what are the things this biotechnology considers? So we all know biotechnology means what? First of all, producing anything which is useful to the mankind by using the living thing, or by using the enzyme, by using the cell, by using any kind of living thing, we are going to produce, or we are going to we are going to produce a protein, or we are going to produce a product which is very much useful to the mankind. The best example I can give here is the insulin. Okay, so to deal with this, first of all, we need some of the tools. Let us consider this as building a house. Okay, if we are building any house, so first we cannot directly build the house. Okay, if I am learning to build a house, I cannot directly go and build the house. First of all, I need to know the tools, right? So which tool is used where, which tool is used for what exactly. Here in the biotechnology also, we need to look into the tools to go with the biotechnological process. So yes, in this chapter we are going to study about the tools. And do you know what are the tools? Can you just tell me in the chat box? Yes, come on. If you know, you can. So firstly, we do require an enzyme. Yes, very good. Enzymes, whatever may be the enzymes. Yes. And then we require a vector. And vector, from the vector, we are going to introduce this, our required piece of DNA, into the competent host. I'm not writing completely. Okay. Yeah. We require a host. So these are the tools mainly we need to know. By using everything, we are gonna see every tool, so we'll understand that in a process. So next comes the process. See, I just want to give you a brief about the process here itself, so that moving forward we do not face any problem. Okay, so what is that process involved in the biotechnology? The process being is that, let me consider any of the example, like a insulin. Let us take the common example as a insulin. Where does this insulin produces in a human body? Any idea you have? Okay, so the insulin is going to produce in the beta cells of the pancreas. Yes or no? Yeah. So it is going to produce in the beta cells of a pancreas, and in this beta cells of a pancreas, we have a DNA. That DNA is actually responsible to produce a protein called a insulin. Do you all know this structural gene? Yes, you have any idea about a structural gene? Come on, say yes or no in the chat box. Manjula, you can also answer. No problem. Otherwise, I'll explain here. Okay. See, if this is a double-stranded DNA, in this, I have this much piece of DNA. Okay. And this piece can produce a protein A. Okay. Only this much part of DNA can produce a protein A. So this piece of DNA can be called as what? It can be called as the structural gene. Structural gene which can produce the, yes, which can produce what? Which can produce a required protein. Exactly, in the beta cells of a pancreas also, there is a small part of DNA which can produce the insulin. So now what we will do in the process, firstly, we are going to take a beta cell. We will digest the beta cell completely and we are going to extract the DNA, the complete long DNA. In that long DNA, by using the enzymes called restriction enzymes, we are going to cut the long DNA into small pieces. So we are going to take our required structural gene or our required small piece which can produce a insulin later. Okay, so that piece of DNA is introduced into the vector so that this vector can, it is going to act as a vehicle. Okay, so and this vector will carry our structural gene into the host cell, and in the host cell we can multiply. I mean, along with the host, our DNA will also get multiplied, so that we are going to get so many host cells. Along with that, inside the host cell we do have the DNA, and that DNA produces the insulin. Later, we are going to collect all the host cell and we are going to digest everything. The insulin we require, which is present inside the host cell, right? So we are going to extract this insulin by the bioreactor. Not only the insulin, the whatever may be the product which we require from that host cell, we can extract that. This is what called as the process. I mean, this is what involves in the process. Is this clear to you? I hope, yes, I'm assuming that you guys have cleared with this. Yeah. So here I'll ask a few questions. The cutting of DNA at a specific locations become a possible with the discovery of. Oh, Manjula, you're here. With the discovery of what? Selectable markers, ligase, restriction enzymes, probes. Yes, correct. So it is by the restriction enzymes. Just now I have told you. If we have a long piece of DNA, and in this long piece, I just want only this much part of DNA. So how can I take that piece of DNA from the complete length? By using a molecular scissor. And that molecular scissor can be called as molecular scissor. That molecular scissor can be called as a restriction enzyme. What about the selectable marker? Any idea? Yeah. So this selectable marker will be present in the vectors. This will be present in the vectors. Okay. So yes. So vectors do have the selectable marker. Based on the selectable marker itself, we can identify our vector. You will get to know about this in the coming slides. And ligase. So what is the help of this ligase? You know, the ligase can be called as a molecular stitches. This ligase is the enzyme which is going to helpful to stitch the DNA molecules. This is what called a ligase. We have, okay. And next is the probes. Yes, probes can be used to identify, okay, if we have a long piece of DNA and we don't want the complete DNA, so we want a pieces of DNA, right? So by using this restriction enzyme, we are going to make the pieces in this DNA, and I want to take out my piece of DNA. I mean, the piece A, piece B, like this. If I have, this is a piece C. So I want this piece A, or the fragment A. How can I extract this fragment A? By using the probes. Okay. So yes, you will get to know about all these things in the coming slides. Don't worry. Okay. Okay, just a minute, Manjula. Can I say that at the end? So why it is called as the restriction enzyme? Why not in a other way? Because see, restriction enzyme can be. Yep. Okay, Manjula. Now itself, I'm gonna tell you this. This enzyme is going to cut at a specific places. I mean, restricted places. Restricted places ಅಲ್ಲಿ ಮಾತ್ರ ಇದು cut ಮಾಡುತ್ತೆ, not in the other places, right? As we know already. Yep. If you want more, we'll discuss just a bit later. Okay. Two bacteria found to be a very useful in a genetic engineering. Experiments are Nitrobacter and Azotobacter, Rhizobium and Diplococcus, Nitrosomonas and Klebsiella, Escherichia and Agarobacterium. Yes, exactly, Manjula. It is a Escherichia and Agarobacterium. So you might have heard about the E. coli, right? The complete name is that Escherichia coli. So this is the, and along with this E. coli, we are going to use Agarobacterium also, mostly in the genetic engineering. Okay. Yeah. So this biotechnology process have been involved, just a minute. It is involved two processes. One is a genetic engineering and next one is a bioprocess engineering. Do you remember the process which I have told you in the biotechnology? Yep. Do you remember the process? Yes. So what is that process involves? Actually, firstly, the process of producing our required protein is considered under the genetic engineering. The processes which do we have to produce. Okay, if I want to synthesize a insulin, so how can I synthesize the insulin? What is the exact method? What is the exact process? Everything can be comes under the genetic engineering. Once we have synthesized the processing, I mean, once we have synthesized the insulin, next we need to take out or we need to purify that insulin with some of the other things, right? That is what comes under the bioprocess. Or you might have studied or you might have heard about the bioprocessor, right? Everything comes under this. So this biotechnology is mainly involves in a two steps: genetic engineering and bioprocess engineering. Okay. And if I want to change anything, I mean, if I have any organism, I want to change genetically. I mean, I want to change that organism in a genetically. Is that possible? What do you say? If I want to change any of the organism genetically, is that possible? According to you, is that possible or not? Yep, it is possible with some of the processes. Yes, very good. With some of the processes. And what are those processes do involve to change a genetic makeup in a particular organism? Firstly, we need to identify the DNA in which we need to, I mean, we need to make a change. Okay, say for example, in the brain cell, I want to change, I mean, I want to change the sequence of DNA, or in the heart cell, I want to change the sequence of DNA, whatever it may be. First, we need to identify the DNA with the desirable characters or with the desirable genes. Okay, and then introduction of the identified DNA into the host cell. You know about a host, what is host and everything, right? Lost a power. It's okay. Can be manageable. You all can see me, right? Am I looking like a host? Okay. So once we have introduced our required piece of DNA, into the host, then maintenance of that introduced DNA in the host and transfer of the DNA to its progeny. Okay. Okay. So maintenance, once we have introduced that our required piece of DNA into the host cell, we need to maintain that required piece of DNA in that host, and later we are going to transfer that piece of DNA into the progeny. Okay, so this is what gonna happen. And do you want me to explain the process of insulin production? Oh yes, Manjula. If you want me to explain this process of insulin production, yes, we can go for it. No problem. Just 15 minutes will wind up. No problem. Manjula is not there, I guess. Okay, no problem. Haha, yes, you are. So can I explain this, Manjula? How exactly the insulin is produced and everything? Okay. Yes, Manjula. I'll just say you about this. See, firstly, as I have mentioned you earlier, what we need to do, I'm going to take, let us imagine we are performing a experiment. Okay, so first what we are doing here, we are taking a human pancreatic cell, that is a beta cell. Especially we are taking a beta cell. Okay, and from this beta cell we don't want a complete DNA, right? So what we will do, we are going to extract the DNA by using some of the enzymes. Okay, the enzymes like, this beta cell is a what animal cell, right? We all know that animal cell does not have any cell wall. So we need to digest the plasma membrane first, because to extract the DNA we need to digest the plasma membrane, and the plasma membrane is made up of proteins, lipids. So we will use the enzymes called the lipase enzymes, proteases enzymes. We are going to digest the plasma membrane of a cell and the membrane of a nucleus also. Now we have got a complete DNA. And then by using the restriction enzyme, we are going to make a small pieces of the DNA. We are going to make a small pieces of DNA. And in that small pieces, okay, let us say we have a 10 fragments. We have got a 10 pieces. Out of 10 pieces, we want only a one piece which can synthesize the insulin. So by the gel electrophoresis and everything, we are taking that one piece of DNA which can produce the insulin. But this piece of DNA is not able to produce the insulin in a human body right now. Okay, there is a one person, he cannot able to produce the insulin. Why? Because this piece of DNA is not able to produce the insulin in his body. So now what we are doing, we both are doing experiment, right? So what we are doing here now, in the other hand, we have taken the bacterium, that is E. coli. Okay, we have taken a bacterium called E. coli. Okay, in this E. coli, this is actually a host. Okay, this is actually a host. From this bacteria, I have extracted a plasmid. Plasmid is a double-stranded circular DNA, we know, right? So we have extracted this plasmid, and by using the same restriction endonucleases enzyme, okay, to get this piece, the restriction endonucleases which we have used, right? The same enzyme we have used here. So then only we can get the similar kinds of ends, right? So we have taken this here. We have cut this plasmid. This is this is a plasmid, double stranded circular plasmid. This is our small piece of DNA. So what we did now, we have separated this. We have cut this, and we have introduced that small piece here. Now you got, right? So now you can observe the original combination of a plasmid is different, and the original combination of a human DNA is different. But here we have introduced a piece into the plasmid. The combination has changed now. The recombination, we have made a recombination here. So this can be called as the recombinant DNA. Is this clear to you? Yes, I guess it is clear to you. Okay. So now from this recombinant DNA, we cannot directly get our insulin, right? So again, what we are doing, this recombinant DNA has been introduced into the host cell. That is E. coli. Again, we are introducing that into the host cell. This is what nucleoid which is present in the prokaryote, this black colored one. and this is our recombinant DNA. Okay, recombinant DNA in the sense here in this condition, the plasmid along with our required piece of DNA. So now what we did, we have allowed this host cell, that is a bacterial cell, to divide by providing a nourishment, by providing a nutrition, everything. We are going to allow this to multiply rapidly. So here you can see we have got, I mean, in a tank, actually what happens, we are going to take, you know about this, right? Making a curd from a milk. What we do there, actually by providing a proper temperature, just a minute. Yeah, sorry. Okay, by providing a proper temperature, and what our parents will do, I mean especially our mom will do, by providing a proper temperature. And firstly they are gonna boil a milk with a high temperature just to kill, I mean just to neutralize the things, right? Just to kill all the pathogens which are presented, and then they'll allow, or they'll allow that to settle to a particular temperature. Once it has reached to a particular temperature, they add some amount of a curd in it. So here by the morning we are going to get a curd. What is that actually? The curd which we add is going to act as an inoculum. Okay, and that milk is going to act as a nourishment there. So by using the nourishment, the complete growth of the bacteria which is present in the curd, that is gonna happen. So that is why by the next day morning we are gonna get complete bacterium in that container where we have gone for the making of a curd. Exactly is going to happen here. We are gonna, there is a tank called a bioreactor. In that tank, it is filled with the nourishment. Okay, so nourishment or the nutrient which is required for that host cell, we will add our recombinant bacterium. I mean a small amount of recombinant bacterial cells, we are going to add that. By the next day or by some amount of time, what is going to happen, we will get a large number of our recombinant bacterium. In that, as we have already produced, I mean as we have already given that with the nourishment, so by using the nourishment, this recombinant DNA will help this host cell to produce the insulin. So now in that bioreactor we have the bacteria which contains the insulin, right? So later by the process called the purification and everything, we can take out this human insulin, and later this is going to test on the animals, on the humans, and everything is done, and finally we are going for the package. Is this clear? Or if it is not, I can explain this in the other way also. So if it is not clear, so if it is clear, we'll move further. Okay. Yep, this is all about the insulin production. Exactly, this is going to happen in the biotechnology. I mean, this is for insulin. We can go for the any other vaccine productions also. Vaccine is also going to produced in the same way. Okay. See, first what we need to do, we need to isolate the DNA. Okay. Yeah. We need to isolate the DNA, and then by using the restriction endonucleases, okay, this restriction endonucleases can be called as the molecular scissors. Okay, so by using this restriction endonucleases, we are going to cut the DNA. The DNA can be of our own, or it can be a double-stranded circular DNA, or it can be a plasmid also. Okay, so here we have different kinds of restriction endonucleases, right? So how can we give the name to them? Yes, here I have one example. Let us take this example itself. EcoRI. EcoR1, sorry, not I. Okay, so if you have an idea, you can say the first letter of the restriction, I mean, just do remember one thing before going to that. All the restriction endo. Okay, am I audible clearly, Manju? Am I audible clearly? Thank you. Thank you. So the first, as I told you, all the restriction endonucleases are extracted from the bacteria. So how we are going to give voices breaking? Let me turn off my video. Yep. What about now, Manjula? See, I told you now clear. The internet is very bad here. Okay. See, first letter of this restriction endonucleases can be taken by the first letter of a genus. Let me tell you, Escherichia coli, E. coli, right? We have extracted this eco R1 from the E. coli. So the first letter of a Escherichia is E. So always, whenever you are giving, I mean, if you have got any restriction enzyme, nobody have given the name to that. How you can give the name? Firstly, you need to take out, I mean, you need to take the first letter of a genus. So this is the first letter of genus. And what about the next letters? These two letters, I mean, the second and third letters are taken from the species, the first letters of the species. See, coli, CO, CO from a coli. And what about this R? Yes, you might have remembered this, right? R strain, S strain of bacteria. Do you remember that? Yep. So exactly every bacteria do have one or the other strain, right? We need to take a strain. Then this is a Roman number based upon the discovery. If it is discovered firstly, okay, from the same bacteria, we have extracted for the first time. Let us give this as a eco R1. If we have discovered the one more restriction endonucleases with the same bacteria again, we are going to give eco R2, eco R3, next eco R4, like that. Okay, I hope this is clear to you. And whenever this restriction endonucleases have work on the DNA, we are going to get the two types of the ends. The first one is the blunt end. Blunt end. And the second one is the sticky end. Sticky end. Okay. So the blunt end means what? If this is the long piece of DNA, and now the restriction enzyme is going to act here. Both the restriction enzymes are going to act at the same place. So now we are going to get exactly the equal pieces, right? Yes, these are the pieces we are going to, this is the piece 1, this is a piece 2, or this is fragment 1, this is a fragment 2. So only the bonds between the two nucleotides have been broken here, not any other bonds. But whereas in the sticky ends, you can observe if this is a long piece of DNA, the one restriction endonucleases is working here, and again it is going to work here. So which means what? We are going to get the small piece and the longer piece, the longer piece and the small piece. So here what happens? The bonds between the two nucleotides will get break, obviously, and the bonds, the hydrogen bonds which are present between the two strands, they will also get break. This gives us the sticky ends. Okay. And you can have the examples of this. Once I share this notes, you can easily get this. Okay. And then is the gel electrophoresis. So in this gel electrophoresis, what do we do? Actually, here these are the wells. Okay, the purple colored lines, what you can see, these are the wells. Once we have cut the DNA into the small pieces, we have this, and we provide electricity to this. Okay, I'm not explaining in a detail. If you want me to explain, just do ask me, I can explain you. Otherwise, we'll just move forward. Okay. Yeah. So once we have poured all the DNA pieces into it, we all know the DNA is a negatively charged. So obviously the DNA is going to be attracted by the positive electrode. But how speedily the pieces can move, that is what required here. The smaller pieces can move very fastly, the larger pieces can move slowly. So this is how the pieces can be separated. Okay, this is how the DNA pieces can be separated. And then we will move all these separated pieces on the nitrocellulose paper, and by adding a probes to it, we can get our piece of DNA. Okay, and okay, this is not required. You can solve these questions. And once we have got our required piece of DNA, then we will go for the polymerase chain reaction. So in this polymerase chain reaction, we need to do, firstly, we are going to separate the two pieces. We are going to separate the two strands. This process is called as the denaturation. And then from this denatured, I didn't get you. Yeah, that's what I told you, right? I'm not explaining the electrophoresis in a complete way. I've just given you the, I'm just brushing up that's it. So if you want me to explain, I can. No issues. Manjula, are there anyone in the YouTube? Yes, Manjula, if anyone in the YouTube, just do let me know so that we can continue the class. Okay. Colgate maybe. Okay. Yep. So once we have separated the two strands, and again to these strands, RNA primer will get attached. RNA primer will get attached. This step is called as annealing. And what this RNA primer will do? RNA primer will add all the nucleotides. RNA primer will add all the nucleotides to it. So here we have got the 2 DNAs again, 2 again. From this 2, we are going to get 4. From 4 or to 8. Like that, we are going to multiply our required piece of DNA by the PCR machine. Okay, so this process is called as the amplifying. And cloning vector and everything can be explained in the next class. So if you're having any doubts, you can ask me about BT cotton and those things also. I'll explain. Okay. Yeah. Thank you. If you're having any doubts, you can ask me. Otherwise, you can leave for the day. Yes, Manjula, you didn't told me yet. Are there anyone in the YouTube? Yeah, Manjula, no problem. No one in the YouTube, right? So can we wind up the class or? Yeah. So yeah, thank you. Bye-bye. Bye-bye, Manjula. We'll wind up anyhow. We have taken the 35 minutes of class, right? So bye. You can stop the live. You can end the live stream, okay? Oof, sorry, in the YouTube. Yep. So can you just ask them to revise? I mean, they want anything to revise. Just do ask them, no? Manjula. Now they have joined after half an hour. Yes, yes. Anybody wants me to this? I can. Voice is breaking. Yes, I'm very sorry. Okay, the internet is not working properly. So then we'll wind up the class. So, yeah, thank you. Bye-bye.